Hello everyone and welcome to another practice session, and today's topic would be bioinformatics, and this topic is a new addition in GATB exam, and many of you guys are really anxious about it. You guys have requested me to take more lectures on that, and I'm really sorry about it because I could not upload much and I would be uploading slowly, so bear with me. Now, you don't have to worry about such questions because the level of bioinformatics that they ask in this exam is rudimentary. And right now, since it's very few days left, what is really important that you should go over the previous year questions. In order to make this venture easy, Unacademy introduces you to the PYQ batch, which is starting from 28 July, where you can start for preparing several exams by practicing the previous year question, including GATB 2020, so you can prepare for your exams better with the help of best educators in India. So why are you waiting for? Enroll right now, download the app, use my code AP10, and you can start learning from the app. So let's begin the question answer session. First question is, which of the following tool works on homology and similarity? Prospect, Emboss, Rasmol, or Blast? So you can guess it correctly, it would be Blast. So we'll talk about Blast, but this kind of questions are really common, and these names especially, which are given in all these four options, are really important. You need to know each of them. Okay, this question can be rephrased like this one. You have identified a sequence which is found in hagfish and speculated that it is similar with human and mouse hemoglobin gene. Which bioinformatics tool would be most useful to understand that? Pattern Hunter, Prospect, Blast, PDB. Again, the answer would be Blast. Why is that? Because Blast would allow you to take an unknown sequence, and then using Blast tool you can compare that unknown sequence and find similarities in known sequence databases. Let's say you have a human sequence database which is already there, or let's say you have a mouse sequence database. You can compare your sequence with that particular database and it would tell you about a match, that too with some scores that would allow you to understand how good is that particular match, what is the quality of that match, and what is the chance that this is a random match. So all of these parameters would be depicted by Blast. So let's go to next question. You have uploaded a query sequence and performed Blast. After Blast, the best search results has a E value of 3. What does that indicate? You found 3 sequences in the database that match your query sequence. E value 3 means chances of similarity is 10 to the power 3. E value describes the number of hits that one can expect to see by chance when searching a database of a particular size. E value does not have any significance in terms of Blast. So E value or expect value is a very important parameter in Blast, and the correct option is option C. So let me tell you what E value is. So E value is a number of expected hits of similar quality that could be found just by chance, right? That means, let's say you have a E value of 10. That simply means that 10 hits can be expected by chance. So the moral of the story, smaller the E value, better would be your match. So you can analyze it from the Blast results. So for example, here one particular query sequence is given, and you see the query sequence where the percentage identity is 100%, the E value is very low, I mean 0, right? Obviously you can see where the percentage identity is 97%, there is some kind of E value, right? So you can understand, smaller the E value, better would be the identity or match. Yeah, you can also see with each kind of result they give you this parameter, E value or expect value. So smallest E value would be the best matched result. Question 3 says, FASTA, FASTQ, SAM, BAM, consider 4 terms listed above. They are sequence file formats, amino acid related information, they are sequence databases, none of the above. So if you get me correct, this particular question is based on sequence format that are used in several databases. All of these names are actually defining some kind of sequence file format. FASTA is very known kind of sequence format that everybody uses, all the database. In fact, in the database you give the FASTA format, so this is very common. Question 4 says, statement 1, the protein data bank is a database for three-dimensional structural data of large biological molecules such as protein and nucleic acid. Question 2 says, statement 2 says, the molecular data available in PDB is obtained from X-ray crystallography, NMR or cryo-electron microscopy data which are submitted by biologists and biochemists from all around the world. Both these statements are correct, only one is correct, only two is correct, or both these statements are incorrect. So you have to choose among them. If you choose correctly, both of these statements are correct. Don't get fooled by this particular word nucleic acid, because PDB also has structural information about nucleic acid and nucleic acid binding protein. Many co-crystallized structures are provided there. Now generally PDB was made as a extracrystallographic database. The extracrystallographic coordinates are provided in the PDB, but nowadays NMR coordinates, cryo-electron microscopy data, their 3D visualization all can be done with PDB. So you can just type PDB in Google and start learning about PDB. Okay, then question number 5 is, which of the following is a protein sequence database? Okay, this is again another question, a very common type of question. So they can ask you which of the following is a DNA information database or protein information database. So you just have to know that which of these are protein information containing, which of these databases contain information about proteins or DNA. So DDBJ or GenBank, both are important information database for DNA sequences, whereas PIR is a database which holds information about protein. Question number 6 says, which of the following tool can provide information about presence of a certain motif? Again, these kind of questions says, okay, if you want to look for particular protein motif in a particular motif in a protein, and you try to understand that what the particular structure may be, then these kind of tools might be useful, and the correct answer would be prospect. So many of the cases what happens, we don't have an extracrystallographic structure for a protein, but you have the protein sequence. So from the protein sequence, using the help of this database, you can understand that what particular motif might be present or might be generated using that sequence. So it would give you a rough ballpark idea that what kind of structure your protein might have. So these are few questions from bioinformatics section, and I would be posting more questions. If you think this this particular session is useful, don't forget to like, share and subscribe. You can also follow me in Instagram, Facebook or in Twitter. See you in the next video. Thank you.